Welcome to part two of mitochondria analysis. And it's been a while since I posted anything about mitochondria, and I've been meaning to, but things just come up and I get distracted. So, to get back to the part two that I promised in my previous video, I'm going to be talking about how you can take the data that you obtain using the tutorial in part one and how to utilize that to then leverage some of the techniques in machine learning, unsupervised machine learning, and some of the lessons that I've learned. I'm going to share some of those with you. So, without further ado, let's dive into this. So, with mitochondria morphology, we all know that mitochondria have different types of shapes, they have different positions in the cell, they have different functions, even, and structure defines function. So, many programs have pre-built designations for mitochondria, such as puncta, large, round, large and round. And then for networks of mitochondria, many designations exist, such as branches, branch length, junction, and those type of things. But there's no real standardization to it. And in my own experience, I've found that mitochondria shapes and their structures, they vary cell to cell. They often vary from tissue to tissue, but within certain, I want to say certain types of cells, these structures are very similar. So why not do something that would help us to understand at scale what different cell types and their morphological features are uh, from the point of view of mitochondria. So to illustrate that point, I wanna take a look at this kind of feature space plot. You see here different pictures of cats and dogs, and there are different features to each. And this example is taken from a Wolfram Alpha website, but it goes to show a very strong point. So if you take all of these pictures and you want to kind of cluster them and separate them, and there are multiple different ways that you can separate these images. So for example, you can do it by fur coat, like I've done here. So these guys are, you can cluster them in or separate them, group them based on off of their fur coat. These middle dogs, these two, you can separate them based on their fur coat, and the cats here down at the bottom, you can separate them based off of their fur coat also. But then you can layer it all together and say, all right, so we have three dog pictures right here, and then separate them again based off of another category, which is the coat color, and then orange cats and their orange fur. And then you get one, two, and three categories of the features that I had presented in this figure, this image. So we can do similarly for mitochondria. We might have a bunch of different cell types all in one data set, bunch of different mitochondria shapes, their structures from either 2D or 3D data. We can then go ahead and separate them based off of their features. And instead of arbitrarily calling them medium sized, large size, networked, and all that stuff, why don't we let the data by itself do the talking and categorizing, so to speak? So, with that being said, principal component analysis with k-mean clustering is a very common approach to cluster data into segments, like you see here with my illustration of cats and dogs. And with mitochondria, things like the size, the volume, the shape, number of branches, total surface area, total count, mitochondria count per cell, and then not to mention if you are doing high throughput screenings for different cell types, then you have that additional feature built into your data set, such as cell types. So using that approach, I'm going to bring up this data set that I have. So this is a data set. Some of this is my own data set and some is synthetic data set that I made up for this example. So over here, I've arranged my data into different conditions. Okay, then I have experimental condition named here. Okay, so these are middle-aged mouse and so on and so forth, younger mice. Okay, so then the image name goes next to it, the mitochondria count, total volume for each individual uh, each each individual cell and the mitochondria that are inside, and then the mean volumes of those mitochondria, total surface area of those mitochondria, and so on. And then at the end, I have this one column that I created called cell type, and I'm designating them with numbers 1, 2, and 3. So 3 cell types, multiple independent variables that are in the exposure condition, and multiple dependent variables, which I'm using as the outcome of the independent variables. So here, these independent variables for me can be anything from drug treatment to any kind of other manipulation that, some people might want to do. Okay, so with that being said, that's the structure of the data that I'm going to use. And then I'm going to use these dependent variables to try to classify or to find underlying groupings of mitochondria that for this data set would allow me to understand or represent for my data set different features of the mitochondria, and then I can further classify them based off of their cell type. So for example, cell type number 1, I don't know, let's call it neuron. So cell type number 1 might be neuron, cell type 2 might be epithelial cell, cell type 3 might be kidney cell or something like that. Okay, so based off of these things, these cell types, then I can further subdivide my clusters and then go ahead and figure out which cell type has what type of cluster that dominates that cell type category. And therefore, now I'm layering categorical approach, but more quantitative driven rather than calling them things like large or puncta or round shape. So by doing some kind of a principal component analysis with some unsupervised machine learning approach, we can get more quantitative outcome and then layer it on top of cell type specific analyses or some sort of other analyses. And I will demonstrate some of that in my code walkthrough when I go through it. In fact, I'm not going to be going through the code itself. I will leave that up to you guys, but I will demonstrate some of the features that I have in there. Okay, so let's move over to that now. So let's get started with the part 2. So previously I showed you all how to obtain data and how to collect, how to do analysis in ImageJ and collect the data. Now what I've done is I've taken many different variables from the way that I did my data analysis for mitochondria and sorted them in these columns so that they're ready for me to use. I've also added in the exposure condition column, and this was done manually to make sure that I don't miss any data points. And then I've also had this column added which tells me which cell types I'm looking at. In my data set, I do high throughput imaging. There are multiple cell types that are present there. And in order for me to kind of keep track of them, I manually annotate the column here called cell type. Okay, you can arrange your columns in whichever way you like. It doesn't have to be in this way, but I'm giving you a general overview of how you can arrange your data to make it work with the file that I have shown here. So I'm going to go very quickly through the first few steps here, and the real part of the part 2 starts here for the PCA, Principal Component Analysis. Okay, so I've loaded in my data. I'm going to go ahead and sort it. This code block created the box plots for me, where I can now see all of my box plots for different cell types here. And then if I wanted to, I could save these as images, which I'm not going to do that. I don't need to save them. I'm going to go ahead and run this correlation, and it will generate this matrix for me, correlation matrix. And all the ones that are less than p0.05 correlations are labeled with these two asterisks like this. Okay, so coming down here to the principal component analysis. So the idea here is to take all of the dimensions. By dimensions, I mean all of these variables that I have, the dependent variables that I am measuring as an outcome of my independent variables, which are my conditions. Okay, so these are independent variables, which are the treatment conditions. And in my case, your case may vary, but these up here, which include total count of the mitochondria, total volume, mean volume, total surface area, and so on. These are all of my dependent variables that I'm analyzing. Now, what I would like to do is take so many of these independent and dependent variables and make them so that I can look at them in a 2D plot while retaining the underlying trends that exist between the dependent and the independent variables. So to do that, I'm going to go ahead and run the principal component analysis, and you can read through this, and there are a number of different tutorials on principal component analysis that are available on many different YouTube channels and also in many different papers. So feel free to read those, but I do try to provide some understanding of what the code is doing in these comments. Okay, so then now that I have created this principal component analysis, what it has done is it has, the better way to explain this would be it has taken all of the multidimensional data where there were multiple dependent variables and one independent or multiple independent variables and squished it or transformed it so that it's easily represented by a component called principal component 2 and principal component 1 while retaining the trends that are present. Okay, so that's essentially what it's doing. It's doing a transformation. Next, I want to find out using this elbow method what is the optimal number of k-mean clusters that I can break my data into and have it grouped so that each individual group represents a certain feature of the data set or the cell type or something else in my data that I can utilize to group my mitochondria data and get some kind of a categorical division going so that I can separate and distinguish the data sets and the underlying trends. So that's what this block is going to do for us. And I don't need to change anything over here. This will output hopefully a nice plot for us to see. So let's run this and ignore these warnings. They are kind of warnings about the number of threads that I'm using on my computer. Okay, so here's the code, right? So that's being run, and it's giving me the optimal number of K clusters down here using the elbow method to tell me that there should be 4 segmentations of my data that I should do. So that there should be 4 different categories or groups of features that I can break my data into. So then I come down here, and because I know this one has given me 4, I will go ahead and change the n cluster parameter here to 4 and run it. Okay, I don't need to do anything else. If you want to save your plots, uncheck these comments here and you will have your plots saved. But in my case, I don't want to do that. You can also save the entire data frame with cluster labels as CSV file, and that usually helps a lot. So it's very helpful to save these things. Okay, so I just ran that and it's telling me cluster 0 features are like this. So there are this many counts of mitochondria in cluster 0. Their approximate average count, average total volume in cluster 0 is so and so on and so forth. Okay, and here's the cluster 1, cluster 2, cluster 3, and that's total is cluster 4. So visually, clusters are separated like this. So 0 is green, cluster 0 is green, cluster 1 is purple, and cluster 2 is yellow, and cluster 4 is this sort of the bluish color. The Silhouette score represents a single measure that summarizes the compactness and separation of the clusters. And I explain over here that it calculates using all the data points and their corresponding cluster assignments. And ultimately, what this is it's a range from -1 to 1, where high value indicates that the object is well matched to its own cluster and poorly matched to the neighboring cluster. Here I have a score of 0.42. And you can see visibly over here too that the separation between the clusters is somewhat arbitrary right now. So the Silhouette score here is not very high. So if it was closer to -1 or 1, that would be the ideal situation. A box plot for each mitochondria parameter in each cluster can be generated using this block number 11. Like this, for example, you can create your box plot for each cluster and the approximate mitochondria count within that. And in my case, I'm using the total volume as one of the parameters, so it separated total volume for cluster 0, cluster 1, 2, 3. And by looking at these, you can kind of figure out which cluster has the highest volume mitochondria. Okay. So these are helpful. And the pair plot for mitochondria parameters, I'm not going to create this in this tutorial. This takes a very long time. But what it will do is it will generate a figure where all of your parameters are drawn out like a matrix. Okay, so it will have it will create matrix plot for all of your data set, and it takes a long time, so I'm not going to run it here. But moving on to a heat map, okay, so this is very helpful to see your clusters broken up like this using a heat map. So here, for example, a red symbolizing a very high number for the branches, number of branches. So very easily I can tell you that cluster number 1 in my case has a high number of branches, and it has a high number of total branch length, and total surface area is also kind of high. By quickly looking at this, we can figure out cluster number 1 is associated with high branch number and total branch length and total surface area. On the flip side, cluster number 3 has low surface area, low branches or number of branches, and low total branch length. So it allows you visual representation to very quickly distinguish different features of your mitochondria that can be clustered as a category. And it allows you to better understand your data and categorize it better. This categorical division doesn't facilitate the cell type here, right? So to now be able to obtain cell type specific clustering scenario, let's move on and cluster our data and kind of find out what are the most common cell types in each cluster. So we get a cell type specific effect. In my Excel file, I have a column called cell type where I have coded each of my different cells that I'm looking at by using numbers. So I'm just going to go ahead and use this to do that. Okay, so this ran and the output is right here. This is a table. It shows me percentage of cells in each cluster. So cell type number 1 is in cluster 0, 1, 2, and 3, and their approximate percentages are listed right next to it. But to be honest with you, I like graphs, so I'm going to go ahead and generate a graph rather than just pure numbers. So to facilitate my my understanding, so I have 3 cell types. I have coded them 1, 2, and 3. And this graph is a stacked bar graph, so showing me that there are about 21-ish cell type 1 in cluster 3. And in cluster 2, I have 61. Cluster 5, I have cluster 1, I have 5 of cluster 1. And okay, let me rephrase it. So I'm more in tuned to understanding data by looking at graphs. Here I have generated a stacked bar graph that shows me how many cell counts per cell type category are present in different clusters. So there were 4 clusters, 0 being the blue, orange being cluster 1, 61 having cluster number 2, and green being cluster 2, and then red being cluster 3. So this visually very easily distinguishes for me how many cells are in each cluster category. And from the data set up here, from this figure, I know that my cluster number 1, right here, cluster 1 has high total branches, number of branches, and high total surface area, and high total branch length. So very quickly, I can come down here and say, all right, so my cluster number 1, which is the orange one, has primarily cell type number 1 in it. So it's primarily dominated by cell type number 1. My cluster number 1 is primarily dominated by my cell type number 1, but cell type number 2 is just one cell in cluster 1. Okay, so I need to collect more data for that one to make it a little bit more robust. But then cluster number 3 was the second, I believe, second highest number of branches. Yeah, second highest number of branches and second highest total surface area. So cluster 2, which is the green one, is again dominated by cell type number 1. Okay, so very easily it allows me to distinguish the cell types and associate those with total parameter change, which in my case is total mitochondria branch count, total surface area, and so on and so forth. Okay, so common cell types in each cluster among exposure conditions. So if you're like me and you're exposing cells to different pharmacological agents and then seeing the effect on mitochondria morphology or mitochondria membrane potential, then this block will be quite useful. So just like before, you just run it, and over here it will plot the exposure conditions. If you are labeling your conditions column as exposure conditions, then you can just run it without changing it. But otherwise, if you're calling them and labeling them differently, then go ahead and change these settings here and change the names to match your own Excel file column headers. Okay, so here this quickly tells me, all right, so ATP has, this many number count. By count here, it means the cell count. So in my ATP condition, I have 6 cells that fall into cluster number 2 and 5 cells that fall into cluster 0. So cluster 2 and 0 are primarily, the outcomes of ATP treatment in my case. Cluster 2 and 0, which both of them have, I want to say, low mean volume and low total volume relative to cluster 1 and 3. Okay, so ATP has that effect in my cells, for my cells, and so on and so forth. So I can figure out which condition is shifting what parameter in the mitochondria, and by correlating those with the cell types here, I can figure out what cell types are most affected by certain condition and certain drug treatment, for example. I've also left in here this Shapiro-Wilks test for normality and Levin's test. Some of you may find this helpful and others may not. Shapiro-Wilks test checks whether your underlying data is normally distributed. So this helps to kind of understand whether you should apply parametric or non-parametric statistics. So I will leave this in here. You can very quickly find the answer to whether you have normally distributed data or not. In my case, all of these are non-normal, so they're not normally distributed. I would need to perform non-parametric statistics. And then next part here, I also do a Kruskal-Wallis H test. Kruskal-Wallis test is for 2 or 3 or more independent variables, and in my case I do have 3 or more independent variables. So this is a non-parametric test to basically identify the underlying trends in your data set. And follow up on this one, you would need, if you were to use this test, you would need to do a post hoc analysis, pairwise comparison post hoc, and that can be done through Dunn's post hoc test. So I'll go ahead and run this just to show the output, what that looks like. So here is the table one that got created. If you want to save this table, you can go ahead and save this as a result, and it will save it as a CSV file somewhere, and you can define that path. Uh, Dunn's post hoc test can be done next. And by the way, these are tests that I'm doing based on my own data set. It may not work for you, so you will have to understand what the limitations of these tests are for your own data set and whether those can be applied to your data set or not. But here I'm providing just a template to help guide. So Dunn's test is a specific post hoc procedure designed to be used after Kruskal-Wallis test. And yeah, it just goes on to tell you a few more details there. But basically, this is what the outcome looks like. So for each of the variables, it will generate a matrix, kind of a table like this, and you can then go through it and see these are the p-values, which ones are here. For example, the control 2 and condition x show significant p-value here. And yeah, these are your p-values for pairwise comparisons. So control 2 is significantly different than my condition x. Okay, well, these are some of the things that are present here. To visualize this in a little bit of a better way, I was preparing two different code blocks, but I think one would just do fine. So the reason I was doing two different code blocks is I was not very happy with the overlap that I see over here with the p-values, and I wanted to minimize the overlap, but these do a fine job, I think. So font size, if I decrease it to maybe like 10, that should work better. Yeah, no, so that's for some of the p-values, because they're so long, there's still overlap here. But overall, there is a way to visualize the Dunn's post hoc pairwise comparison. The last thing I did over here was another visualization like this, and feel free to use either one of these, but this is just to help guide the analysis. Okay. So I will share this on my GitHub repository. Feel free to utilize these. And the last thing I will mention here is that sklearn, this library has been deprecated, I believe. So I may at some point update this, so do check back in on the repository to kind of get the most up-to-date script. It's easy enough to download and run this in Jupyter using your Anaconda environment. So you can create an Anaconda environment with all of these libraries and go ahead and run this. And if you followed the tutorial 1, you will already know what to do. This part here is where the mitochondria analysis part 2 really starts to come out. Okay, well, that's it for this tutorial. Thank you so much. Have a nice one.